The overall goal of this procedure is to study the three-dimensional movement patterns of zebrafish or other aquatic animals and to analyze their kinematic, spatial, and social characteristics. This allows detection of altered behaviors due to genetic mutations or chemical and environmental factors. This is accomplished by first calibrating the system, which also corrects for the sizable image distortion generated by refraction. The second step is to record video frames at relatively high temporal resolution from at least 2 different angles simultaneously. Next, the software reconstructs the trajectories of the fish. Advanced algorithms are applied to reduce the effect of noise and to infer the position of a fish when occlusion or clustering occurs. The final step is to analyze the data and to perform statistical analyses for velocity, acceleration, turning angle, horizontal and vertical distribution in the observation container, inter-individual distances, and other parameters of interest. Ultimately, the high-speed calibrated 3D tracking is useful to study the behavioral deficits in mutant zebrafish and the effects of drugs on a variety of behavioral systems for the purpose of drug discovery.
One advantage of the system is that it allows studying behavior of fish in 3D. This is important because fish naturally move in all 3 dimensions. And when recording multiple fish simultaneously, 2D recording